feeling like sunshine, like springtime, like something's in the water and I'm taking a deep dive. I'm feeling so weightless, like I'm gonna make it, and nothing in the universe can take this. I can see it clearly now, nothing gonna bring me down. I got my head held high, I'm on my way, and I'm following the signs to my happy place. I'm walking in the light of a brand new day, and I'm right where I belong in my happy place. I got my head held high, I'm on my way, and I'm following the signs to my happy place. I'm walking in the light of a brand new day, and I'm right where I belong in my happy place. And I'm right where I belong in my happy place. And I'm right where I belong in my happy place.
Hello and welcome to the National University of Singapore Yonglu Lin School of Medicine Healthy Longevity webinar. Thanks for joining us again. We have Professor Gerd Kempermann tonight to talk about the aging brain a little bit. But before we get to that, I want to remind you to use the Q&A function to send questions. We're excited to hear your questions tonight. And also, I want to introduce Shija Navakode, who's a senior research fellow here at our very own Center for Healthy Longevity, and she will talk about why females are more vulnerable to Alzheimer's disease.
Thanks for the introduction. As mentioned, my name is Shija, and I will be talking about why females are more vulnerable to Alzheimer's disease. Or to put it more simply, why females are at higher risk of developing AD compared to males. Alzheimer's disease is a progressive neurodegenerative disease characterized by excessive accumulation of amyloid beta plaques, neurofibrillary tangles, hyperphosphorylated tau, along with synaptic and mitochondrial dysfunction, dysregulation of calcium homeostasis, and neuroinflammation. The clinical symptoms of Alzheimer's disease typically include progressive loss of memory that disrupts daily life, along with other behavioral complications. If we look at the risk factors for AD, gender plays a significant role, with women being more susceptible to AD than men. Approximately 1 in 11 males and 1 in 6 females have the chance of developing AD over the age of 65, thus indicating that almost 2/3 of AD patients are women. The aim of my work is to examine whether males and females show differences in synaptic plasticity and memory in AD. Synaptic plasticity refers to the ability of the synapses or the ability of the neurons to modify the strength of the connections according to novel experiences. So the next question would be, if such a difference exists in plasticity and memory, does that contribute to AD pathology? In order to study this, we used a mouse model of AD called APP-PS1, which contains human transgenes for both amyloid precursor protein and presenilin-containing mutations that are associated with early onset AD. For the experimental part, we used a cellular correlate for learning and memory called long-term potentiation, or in short LTP, which can be induced in hippocampal slices using different protocols like strong tetanus, theta burst stimulation, or population spike timing dependent plasticity. Coming to the results, here is a schematic representation of a hippocampal slice with electrodes located. S1 and S2 represents 2 stimulating electrodes placed at independent synaptic inputs to a single neuronal population from where the recordings are made. In the wild type males and females, the theta burst stimulation induced a persistent and long-lasting LTP, which was stable for 3 hours. And very interestingly, in the Alzheimer's disease mice, in both males and females, LTP is impaired. And more interesting is the fact that the female APP mice displayed a faster decline in plasticity. It suggests that the synaptic strengthening processes are affected in female mice with AD pathology, potentially contributing to the cognitive deficits that are associated with AD. At behavioral level, we studied associative memory using behavioral tagging paradigm. We used an inhibitory avoidance task where the mice is placed on a platform, and when they step down, they receive a weak foot shock. And the memory was evaluated by comparing the step down latency at different time points after training. The results shows an impairment of associative memory in APP-PS1 mice compared to the wild-type male and females. So in line with the plasticity experiments, associative memory is worse in females compared to males at 1 hour, 24-hour, and 7-day post-training. Our transcriptome analysis indicates a robust upregulation of neuroinflammatory genes and downregulation of memory-related genes in APP-PS1 females. This difference could serve as a molecular basis for the greater impairment in the long-term plasticity and behavior in female AD mice. And lastly, we performed immunohistochemistry using antibodies that are targeted against A-beta and microglia. And the quantification of the A-beta plaques and IBA-positive microglia indicated that the female APP-PS1 mice have an accelerated A-beta pathology with an increase not only in the plaque burden and plaque size, but also in IBA-positive microglia in the hippocampus. To conclude, the AD females shows a faster decline in plasticity and memory, and for future works, this calls out for a gender-based approach for intervention studies in AD. And last not least, I would like to thank Saji, Toheen, George, and all my colleagues, Maria, Vibha, Ria, and Isong, who had been part of this work. And thanks to MOE Tier 3 grant for the support. And finally, thanks to all of you for the kind attention.
Thank you, Shija. So now it's my pleasure to introduce Gerd Kempermann, who studies the neurobiological foundations of how behavior shapes and individualizes the brain. He's a professor of neuroscience at Technical University Dresden in Germany and heads the Dresden site of the German Center for Neurologic Diseases. The title of his talk tonight is Neurobiological Mechanisms of Individualized Aging. Thanks for coming on the show, Gerd.
Hi, Brian. Thank you for having me today. And I'm looking forward to, first of all, presenting and then discussing with you. So this talk is about individualization of responses. And it's not so much about aging, as you will see per se, but about a very fundamental mechanism that has a large impact on our thoughts on how aging is different. Normally, in science and sometimes in real life, we consider individual behavior as a nuisance. It just falls out of what is expected. And when we think more about it, it's actually true that we all want to be individuals. We want to be respected as individuals, and we don't want to be average. So it's important to look at differences because they are important in science, and they are particularly important in what we are all interested here in, and that's the question why and how people age so differently, and the extent to which our own actions actually contribute to that. And this concept of the reserve, the neural or cognitive reserve, or however you might call it, implies that there are different causes that together make up a range for compensation, which is called the reserve. And the larger our reserve, the better we can compensate the problems that come with aging, and the greater our resilience to neurodegeneration. But the question is, how does that actually come about? What is this reserve? And that has been a question that has been addressed from many different angles. And the question is how you can study something like that in rodents to make it accessible. And the paradigm of choice for many of these questions that look at activity, at environment, is the so-called enriched environment. And you can see here mice running around in a large cage, having a lot of fun, and it's more fun than in the standard cages that we normally see, or the mice normally see. And this paradigm is very old, has been used in many contexts for decades now, And the general observation is that you see huge differences between just these two conditions. So if you have the phenotype P of interest, then so the assumption goes that because we keep the genetics constant in this paradigm, we use inbred mice or rats, then we expose the environmental factor. And this has been a very powerful paradigm, and a lot of insight that we have about the impact of environmental factors on any phenotype, including aging phenotypes, has come from this paradigm. But there is a certain limitation to it, and we have become interested in that. But first, let's have a look at what exactly can happen, the extent to which the brain becomes different in such treatment. And I'm showing you here data from a collaboration with my colleague Haider Amin here at our institute. And on the right-hand side, you can see a little microchip. It's a CMOS multi-electrode array. And below, you can see that on such array, a mouse hippocampus nicely fits. It's an area about the size of my pinky nail, and so 2.6 by 2.6 millimeter, and a nice hippocampus, a mouse hippocampus nicely fits on that. And you can see that on the left, how it looks when the section of the hippocampus is placed to it. On the next slides, you will see what happens if we have a mouse that is has been living in a standard environment for weeks, and you see local field potentials. You see excitation. This is sort of spontaneous activation. There's no activation from the outside. You see. And you will see the video again, that the excitation starts here, in this case, in an area near CA3, CA1, and then spreads to the entorhinal cortex, which is labeled with EC here. So see that again. And on the right side, you will see the same preparation with the hippocampus from a mouse living in an enriched environment. And this runs in parallel, and you can see that there's much more going on. There's way more going on in the enriched living animals. And again, the only difference is that they have been living in a different environment for a long time. And if you take this data and look at them, this local field potential, just the rate, you can already see on the graph on the left side that there are huge differences. And there's also differences in variability, as you can see from the arrow bars. In all of the regions of the hippocampus, you see that there's more of these activity and that there's greater variability. And on the right, you see, if you plot the network that you can deduce from these activities, that there's a much richer network in the enriched living animals. So the electrophysiological properties of the hippocampus are completely different. They're completely rewired just because these animals have been living in an environment that exposes them to more friends, to more toys, but otherwise nothing has been happening. There was no intervention on the side of us as examiners. And these networks that we see here, they have particular properties. Like many networks in biology, they have properties that you all might know from airline networks. And these properties are called small world networks. And small world networks have particular properties in the sense that they have short paths between distant nodes. So if you go from the East Coast to the West Coast in the US, no matter where you get onto the plane, because the airline has set it up such that you can use their hubs here. This is Delta Airlines. You can go through Atlanta, Salt Lake City. And usually with one or two hops, you can make it from even a small place on the east to one in the west. And these properties that rely on these well-connected hubs and have these particular path lengths, they are called small world networks. And many biological systems have these, and they are the dentate gyrus network appears to be one of these. And if you have this exposure to enrichment, you see that this network becomes more complex. And I'm not going into the details of these network properties. They're always on the y-axis here in this graph. But what you can see is that essentially, no matter what you're looking at, you see that there is an increase in this property the networks are better connected. And there's more redundancy in the network. And the path length, that's the only thing where the opposite picture is seen on the lower left, is shorter. It's dramatically shorter. So these networks are much more efficient. Now, if you use these networks that we have derived from these data and sort of run a simulation on them, you can also play with something that is similar to neurodegeneration or aging, if you want, because you can randomly remove or have the computer remove nodes from these networks. And then what you see here is that when you have removed from that network 550 nodes, the standard house network is gone. There's no ability of the network to process information anymore. Whereas in the enriched case, there's almost no effect. The network is completely stable. It's more robust. And that tells us that these complex networks, and again, the complexity only arises from the fact that these networks obviously have been used more because the mice were living in an environment that was more complex, that induced more activity on their part. These networks are more robust against change. So if we come back to this model, or this general concept of the model, then the idea was that this paradigm would expose the influence of the environment. But the question is this really all? Is it true that we only control for the genetic background, the rest is here the environment? So I'm particularly interested from my own scientific background in a type of plasticity that we find only in the hippocampus and only in the dentate gyrus of the hippocampus, which is adult neurogenesis, the production of the lifelong production of new neurons. And this production of new neurons is, as you have seen in the past already, very much activity dependent. And in an old study that I've done when I was a postdoc with Fred Gage, we had seen that when you host animal in such an enriched environment, in the hippocampus, you get actually more neurons that are born in the adult. And so these neurons, as we know today, they play an important role in hippocampal function. And so when you expose animals to such environmental enrichment, you induce some structural change in the dentate gyrus, in the hippocampus, and we have seen that this changes completely the network, and it also changes the network at the level of the nodes. There's a few more neurons that make up the new connections. So a very profound change. But in the many experiments that we have done over the years with this paradigm, we have always wondered whether there might not be more. And in the many studies, we had an observation that I'm showing you here, and that we have then in 2018 finally really nailed in a study that was specifically designed to bring this home. You see in the third panel these new neurons, and you see control and enriched animals, and in yellow you have the enriched. And you see again the effect that we had seen back in 1997, that exposure to an enriched environment induces neurogenesis. You have more neurons. But what you can see here also is that there's a way greater variance. So a little bit like what we had also observed in this electrophysiology study, the animals in the enriched environment seem to be more different. And while there is a mean effect, there's also an independent, or only partly independent effect on the variance. And you can see that is also statistically true. There is an F-test, which is like a t-test for variance. And so exposure to enriched environment induces variability in this response. And this variability, we reason, might be an indication of individuality. The mice become more individualized in their response to this environment. And that makes this paradigm slightly different than what it was before. Because if we think about these mice are all living in the same cage, and if we not so much focus on the control group that lives in another cage, we can also argue that within that cage, the animals live all in the same environment, so we effectively control both genes and environment. So if we see differences, these differences can only come from what is called the non-shared environment. And the non-shared environment is simply speaking, activity in the sense of behavior. So to really use that, we had to use a different type of enriched environment, which is Buckingham Palace style enriched environment. Buckingham Palace has 700 rooms. Our cages have 70 rooms. You can see here they are all connected. A classical standard rack. You see the mice, and you can see that the cages are connected, and the mice can essentially run through all 70 cages. And whenever they pass through these holes, they give a contact via a little transponder that is injected under the skin in their neck, and at all these connecting tubes, we have ring antennas. And so if we house animals in this cage, we have information about their whereabouts and their time, when they change the cage, and of course their identity, 24 hours a day, 7 days a week, and we can run that for months. And so we get very extensive data sets about the behavior of these mice, and they start early in their life, and they start together in that same environment. And what we then observe, and these are curves that are a bit smoothed just to make it a bit more obvious, because there's quite some variability on short range. But if you flatten the curves here a little bit to make it more intuitive on a figure like this, the math can of course be run also on the other data, then you can easily see that there is a certain dispersion of these trajectories. When you start at the beginning, the curves are all together quite closely. When you move at a later time, at the end of the experiment, half a year later, you can see that the animals are much more different. Some are way lower than others here. And you can also quantify this, and this has been done in many studies that we have run by now. And you can always see that over time, there's a certain habituation. The animals become a bit habituated to the environment. It's not a very steep decline, but slightly. But more importantly, at that same time, the variance increases. So these behavior trajectories, these individual trajectories of activity, they diverge. They become more different over time. And it's a natural thought was, of course, to relate that to adult neurogenesis. So is there a relation? And at least here in the 3 studies that I'm showing you here, you can see that there is a correlation. It's not excessively strong, but it's, we thought, surprisingly strong, given that this is just one readout of many, between this activity, which we call roaming entropy, a probability that a mouse is found at a given place at a given time, with this level of neurogenesis. So the individual level of activity over time in that cage has some predictive power for the level of brain plasticity as we see it at the level of adult neurogenesis. And in an unpublished study, to some extent, also in electrophysiology, but there's a bit more challenging to demonstrate, obviously. Now, there's even some causality in there, because there are mice around, cyclin D2 knockout mice, which don't have adult neurogenesis. They have normal neurogenesis during development, and also outside the hippocampus throughout adulthood. But for some reason, the redundancy for the cyclins is lost in the stem cells that are in the hippocampus. And so these mice have a normal dentate gyrus, and they have no adult neurogenesis throughout their adult life. And you can see on the left here that the wild-type mice, they have the same pattern as we have seen it before. There's an habituation effect, and there is an increase in variance when they are exposed to these cage systems. But if there is no neurogenesis available, then this trajectory disappears. Then these mice have show no habituation, and they show no variance increase. So they stay more active in a way, but within that activity level, they are, we think, less more rigid. They don't respond to the exposure to the enriched environment with this increased individuality. Now, finally, we had already seen in the past that exposure to an enriched environment has an effect on connectivity. And in the hippocampus, we had seen that there were structural changes, and we had seen that there were indications of this individualization on the network level, and there's in the hippocampus, and that there are these individual behavior trajectories that correlate with levels of structural plasticity in the dentate gyrus in terms of adult hippocampal neurogenesis. Now, what happens beyond the dentate gyrus? What happens beyond the hippocampus? And here we used a structural MRI on our mice, and this is a collaboration with Anthony Vernon at King's College in London. And the experiment was done the same way as I've shown you before. And if you just take the enriched, the individuality cage as an enriched environment and compare it to a group of mice that are housed in standard laboratory cages, you can see that there is gray matter changes, gray matter volume changes in the hippocampus. That's sort of expected. The hippocampus seems to respond very readily to all kinds of stimuli, and you can see that also in mice and with the MRI. Now, the question is there also an indication of things going on outside the hippocampus? And if so, would these also show some correlation that, or some indication of that there is an individualization effect? And if you do that calculation, so if you use the slope of the habituation as indication of these individualized trajectories, you can see some correlational patterns, but it's very hard to interpret because you see there's something all over the place, and it doesn't give you a clear picture. So we used a different method, and this was by registering these data to a atlas, and used the correlation in change or the variation as between these different regions as a measure. And you can see that the correlation is relative. There is correlation between regions in the standard housed animals, and it depends really on the region. We don't go into the details of what is what here. We just look at this pattern. And what you can see is that this correlation, the correlatedness, increases dramatically when you look at the mice in the enriched environment. So the brain regions become more correlated in their volume. And what correlatedness actually means is, of course, hard to understand, but it is very similar to a concept that in fact is used in humans and human MRI studies, which is called structural covariance. And all of MRI data that deal with the question of the connectome as it is accessible in humans is essentially based on variations of this idea of that regions that correlate with each other are those that are also more connected. So now, how does that relate to individuality? Because you obviously cannot do correlation with an individual mice. You have to stratify them and use here the top and the bottom of the mice living in the enriched environment. So this is the signal that we have seen in all mice. And if we now look at those that stay on top of the spread, you can see that the correlation is even stronger. And if we compare that to those that live in the enriched environment but are at the bottom of the spread, you can see that those with its downward trajectory, they have a very different pattern. And this pattern is very similar to the mice that have been in the standard cage. So we have indication that the connectome in the mice, at least by the standard of structure covariance, also changes when exposed to this enriched environment, and it changes such that there is good indication of some individualization, as far as we can show this with this stratification. So with that, I'm coming to the end. I would like to summarize for you that behavioral activity in response to an enriched environment can trigger widespread network changes in the hippocampus and presumably beyond, and that such networks are more robust and resilient to change, to degeneration. And individuality even emerges if you keep the genetic background and the environment constant. And this, what is then called the non-shared environment, shapes the connectivity throughout the brain. Individual regulation of adult neurogenesis thus, and this plasticity thus can be linked to individual behavioral trajectories. With that, I'm at the end. These people from my lab over the years of the lab reunion we had last year, the studies that I'm showing you also spend some time, but I would highlight Jatna Bogada-Lopez and Sarah Zocher as the leads students for the project, the main project I've shown to you today. At the bottom, you find our funders, our collaboration. The main collaborators I've talked, whose work I've talked about today was Haida Amin and Anthony Vernon, and the concept of roaming entropy has been developed together with Andreas Brandmeier and Ulman Lindenberger. And with that, I'm at the end, and I thank you for your attention today. I'm happy to take questions, discuss with Brian.
Thank you. It's a fascinating talk. Lots of questions. Maybe I'll start with one I was thinking about, and maybe I'm sure I can articulate it perfectly. But you talk about genetics and environment. You can talk about shared and non-shared environment. This like simple like things like stochasticity come into play to define individual differences.
Absolutely. Absolutely. There's this is a big discussion in this field of individuality. And the extreme thought is it's all stochasticity, at least initially. And the question is that extreme position really true? So we at least see that over time there are correlations actually with the trajectories that emerge, and the trajectories don't emerge exactly on the first day. So it's presumably a little bit more than just even at the beginning than the trajectory, than the stochasticity. But stochasticity certainly plays a role in setting mice on a certain trajectory. If you can imagine the mice, they first meet whom they talk to at the water bottle. This might then put them on a track for a certain behavior in the future. There's a social effect in here that we are also studying in other experiments, and this will be triggered as in humans by random effects, whom we meet, how our mood is when we meet somebody, and these things. It goes down to the level of stochasticity at gene expression levels, which also play a role, presumably, because this has gone to the cellular level. And you can imagine that if a cell doesn't respond, that might be due to stochastic effects. But that gets very, very difficult to study. But I think it's a very important point that you're bringing up.
That's a good question, how you try to measure that. You brought up another topic, though, which I was going to ask about, which is sort of, we know with aging that social connectivity is associated with better cognitive function and health generally with age. Can you measure the social connectivity of the mice in this?
Yeah, we're actually doing that, and these are ongoing projects. I didn't really want to get into today, so if you invite me again a couple of months down the road, I can talk about that part. This is ongoing, but it's clear that there are effects of social interaction, and that you can even in this from these antenna data, you can get to a lot of information about social structure. about hierarchy and about interaction, and this it will also explain, besides this fact of this roaming entropy that I showed today, part of this variation.
Yeah, I was thinking during your talk that I'm not going to show your talk to the mice in my lab because they'll all move to your lab. It looks like a lot more fun.
Yeah, we have a lot of mice knocking on our doors that they would like to pet.
Our advantage though is we keep them alive forever, so they can have a long, boring lifespan or or a very enriched lifespan. It's a tough choice. I
Yeah,
get back.
a lifespan study yet. That, of course, is something that is interesting to see whether this individualization keeps going and what the impact then on really aging is. We start usually very young because we don't want to have studies where there is a lot of interference of earlier exposure for obvious reasons. That's, of course, is interesting by itself, but it's a different question. So there's really long-term studies we haven't done yet.
Yeah, I think that I was thinking of it in terms of just, well, it's a hard. I was thinking about this. For one thing, let me go back to stochasticity first. I want to come to that question too. Because, one of the challenges we have in the aging field, and this is true in genetically identical mice, it's true in genetically identical worms and genetically identical yeast for that matter. When we measure the survival of a particular strain, even though there is similar conditions as we can put them in, there's a large variance in how long these animals live. Some of them die quite early, and some of them live almost twice as long or more. And no one has ever really explained that phenomenon well in the context of aging.
Not to knowledge, and it's one of these interesting questions that really goes to this question of the non-shared environment, or how you want to call it. So yeah, the can of worms, they're all in the same environment, and even there you see some variation. Again, there is some stochasticity, but I think it wouldn't be good to restrict the hypothesis just to the stochasticity part.
I didn't mean to do that.
No, I'm aware of that, but I mean, it would be an extreme position, right? That you say, well, it's all genes stochastic and the rest is stochastic.
Yeah.
And we think, no, it's not all. There's also behavior, and behavior has the components that we have already talked about, which is just this motor activity, the social. But the other factor that comes also from the literature on enriched environments, of course, there is a learning effect involved, right? So you shape behavior by past experience and by prediction of future experience. So if there is, for example, a very dominant mouse in the cage, then you learn that you better avoid her or him, and you don't want to go in that particular cage. So behavior is also changed by learning. So these are three factors. The fourth, that is in the historical literature, is just the sensory enrichment, richness, I want to say. And that also introduces a difference that's more on the true environment part. Though, if we assume that the mice see the same thing, or the worms or the flies, which might not even be true, right? So there might also be just sensory differences, which we would have to look at.
Yeah. Coming to the aging, it's been shown that enriched environments, there have been limited studies, but they show that enriched environments for mice extend lifespan generally. That's not the complex cage network that you're looking at, but it's the simpler system that you showed at the beginning, or something more equivalent to that. So that's always been fascinating. And I wonder if neurogenesis has some role in the longevity of these mice.
That's a very good question. And if we assume that the longevity in mice that are exposed to nourishment has these four factors, then we would assume that learning plays a role. in this fitness for age. And it would be consistent with many circumstantial and studied observations in human, right? Where we know that education and training play a certain role, and that they are not isolated, that when you have that cognitive fitness, it usually comes along with other fitnesses as well. So there is this shared core mechanism which we don't really understand, and vice versa. We know that physical activity alone triggers already some benefit for cognition. So these are all not unrelated. So, and that this also seems to converge to some extent, not fully, but to some extent in the hippocampus. So I would think that this is an important region to look, but I'm not saying that it's the only region and that it explains everything and the truth is in the new neurons, which is an interesting side thought that there might be something in there, but it's clearly part of a big puzzle.
You touched on this already, so I don't know if you've done this experiment, but I'm quite curious if you take, you don't even not even aged, you take animals that are used to a standard environment and put them into an enriched environment a little bit later after development. Or maybe interestingly, the opposite direction, take them from an enriched environment into a standard environment. Maybe in that latter situation, does the activity in the brain change or go back, or does it?
I haven't done it, but we did this withdrawal experiment, and there's very interesting observations. So first of all, it's good news that things that the early effect doesn't completely go away. So there is a benefit that is maintained. But of course, the acute effect goes away. So because there is no continuous stimulation, there is no exposure, and so there is a drop in these individualization curves, which is fairly well. It was actually expected. What we didn't really expect was that the early effects were so stable, and this included very stable effects at the epigenetic levels and clustering on genes that are involved in plasticity. So it seems that when you have the situation that you describe of with this withdrawal, you have a situation where the potential for this plasticity remains high, which speaks to the recommendation that if you do something early in life, that's actually a lasting benefit. But then it's not used anymore because there is this exposure and this activity is missing. So this is something we really would love to understand, and that's actually also the link, one of the links between our research and the longevity and aging research proper, as you and many of the other people that you have in this series and in your center do. Because this is, at this point, these fields then link, and you can bring these research interests together. So we're coming sort of a little from the other side. We're trying to understand this profound mechanism. And then it can be applied to the much more cumbersome to study longevity pair, which takes several lives of PhDs to conduct this study.
This reinforces my belief in the true value of the university system. It creates a crazy enriched environment for people to be in for a few years, and that drives the individuality.
I mean, science is always there to show things that we have known all along.
You mentioned cyclin D2 and be involved in neurogenesis, and that reduces the variance in the assay. Are there other genes that affect other processes in the brain besides neurogenesis that can alter this variance as well?
Well, the cyclins we use, because it's a genetic model of doesn't have neurogenesis. It's not a perfect model because the cyclin D2 is involved in other things, and also there is more redundancy. This is not perfect. But other models of ablating adult neurogenesis are worse, because if you irradiate, for example, that's a standard paradigm to get rid of new neurons in the adult hippocampus, you get also inflammation. And because we study our animals for such a long time, this is a real problem if you have all kinds of things that you cannot control. The same is also true for many of the genetic models that are involved. There are many models based on stem cell genes and on selective knockouts, but the recombination is never perfect, so you never know what you have, and there are rebound effects. So this is why we cannot use that, and that's why the cyclin came in here. That perhaps I misunderstood your question a bit. Perhaps you were...
That was one part of the question, but the other part was, what about the genetic mutations or even pharmacological interventions that affect certain behaviors or
the only studies that we have really done, one is also published, have been using a knock-in model of Alzheimer's. So the student talk that we heard was with a classical overexpressing mouse model. And this is a knock-in model, and the knock-ins have the property that the AD phenotype comes from a promoter, APP promoter, that is the endogenous one. So the regulation is maintained. And that, of course, makes a lot of sense if you're interested in behavioral effects on the pathology, because if you have a very strong promoter that overrides any physiological regulation, you only look at compensation, but never at the pathology process, because the regulation is gone. And so if we use these mice start getting a phenotype at about 8-9 months. And we did our experiment well before. And you could already see that these mice in their behavior become more rigid. They are less individualized. Their behavioral range was already reduced at a time when they were completely healthy by all the standards of the literature and of the models. There were no plaques, there were no behavioral. If you tested them, there's no real effect. So this is a very subtle change, that they already started to behave slightly differently, and these trajectories are reduced. So this is so far the only model where we have seen that, but I presume that you can show similar things for other models, especially of neurodegenerative diseases, and those that are related to aging phenotypes. I predict if you would interfere with the insulin pathway or something, you would see such things. But it becomes messy, and that's the problem, right? You interfere with so many systems that it becomes harder to find the good readout. And our advantage with this model is that we have such a specific readout that we can really put nice numbers to it. But we're not saying this is the thing.
Let me bring in Kalisha, because there's a number of questions from the audience.
Hi, Kalisha again.
Hi, Professor Gerd. Thank you for your presentation. It's really nice to see happy mouse in a big cage. So we have a few questions here from the audience. First of all, what are some examples or applications of such non-shared environments in humans that we can do to deter Alzheimer's disease? Could it be like reading and exercise and social relationships?
Well, yeah, I mean, there's this non-shared environment. If you really want to be bold, you can relate it to what we call lifestyle or so. So it's the how we live rather than the fact that we live and that we are determined by some environmental factors and relate to genes. So the choice is yours. I mean, these are mice, and so we are really careful in extrapolating too wildly, but the background of our research is, of course, to understand what lifestyle actually means. I mean, in humans, this is something that is a very broad term, and it means essentially everything. And to understand what biologically happens if you change behavior, this is very difficult to study, because it always is, in humans at least, intermingled with this genetic and this shared environment. And so our paradigm has a nice advantage that we control both of these other factors and expose this. But it's very hard to make this step. But your intuition is right? This is the direction we want to take it.
Okay, thanks. So the next question is, any thought on how the lower strata mice have connectomes similar to the standard conditions?
How the what? Sorry, I was...
Any thought on why the lower strata mice have connectomes?
Lowest.
Yeah.
Yeah. Well, this is intriguing. They didn't much differ from control. So it seems that the exposure to the enrichment, the classical way of looking at that, is not enough. Because what really plays a role is how the mice respond. And this is different. and they are on different trajectories for their response. And what exactly is happening? I mean, this is exactly the interesting question that arises from our studies now. What's going on here? And we have looked at limited things. We have looked at neurogenesis. I have looked a little bit at connectivity in the hippocampus. We have done this MRI study. We see there are changes. They correlate. The causality in the sense on what mechanistically happens there. This is something that will keep us busy for a couple of more years in science.
All right, this leads nicely to the next question. How do you know that enhancement of the neurogenesis effectively leads to enhancement of connectome?
We don't.
You don't.
These are two different experiments, and of course we know that on a micro scale, if you have more neurons, you also have more synapses, because there are more neurons that can have synapses. You also have an axon. So at the level of the connection between dentate gyrus and CA3, you clearly have a change in the connectome. But the connectome actually is not one fiber tract. The connectome is this whole thing. The assumption or the hypothesis is that this change actually triggers the downstream changes. But it's not everything. That's for sure. There will be other direct effects as well. But you can imagine that if you change the input to CA3, you change the input to CA1. You change the output to the subiculum and the parietal cortex, and you might change how the input to the hippocampus is. So it's easy to assume that there might be a causality here, but that we couldn't show yet.
Is it something that you will be looking into? The effect mechanisms? Okay.
Of course. It's pretty obvious, right, that this is a very interesting and important question.
Mm-hmm.
And the observation that this individualization to some extent, only to some extent, but to some extent depends on the new neurons, suggests that the behavior, the downstream behavior, depends on this plasticity in the hippocampus. And so that there is something like a feedback loop, that if you don't have a functioning hippocampus, you change the output and the output behavior. And so you assume that there is a link between the structure, the function in the hippocampus and the rest. But clearly, this is not explaining everything, and this is not the whole story. It's just one intriguing part of the big picture.
I see. Okay, moving on to the next question. Was social hierarchy or gender differences in your mouse studies studied? And how did it correlate with individuality and connectome studies?
Well, as we said, we are doing the study on the social effects. They are there, but this is something for a later talk and upcoming papers. Yes, there is some interesting effects. The sex differences, well, you have to do separate studies on that, because this is a special way of doing enriched environment, because if you have mixed sexes in such a case, you can imagine that numbers change quite dramatically over such a long experiment. So these are different experiments. Doing these kind of experiments with males is challenging because they are territorial, so you need more space. But you can do it, and by and large, the results are very, very similar. The effects are a little bit more exaggerated because you have more of this. Here we know that the social structure is very different. But it's the same thing, but a bit more exaggerated. But it's interesting, in our study, this is all with mice, with female mice, and the female mice who are supposed to be more similar in general also show this strong effect as I presented today.
You may not have done this, but I'm curious about a model for autism in this study, in this kind.
I haven't yet done that. It's a great question. It's a wonderful question, and it makes a lot of sense too. But we first wanted to get a grip on the social structure, and this before we embarked on a social on a model that is primarily characterized by effects on social behavior, or not primarily, but also effect on social behavior.
Alicia?
Okay, next question is, do you see a difference in the social interactions with time as the mice age?
Well, again, the social studies are ongoing, and so as I don't know yet that fully. I would presume that there are changes with age. We also see in the baseline studies we see changes with age. So the longer studies that we have run have been almost 8 months now. So that for some people would qualify as an aging study, but not probably in an aging center.
No comment.
8 months is not really old, but it looks at a life course, at a substantial part of life course. And then you see that these trajectories, they just don't go on diverging forever. These curves, they start to flatten out a little bit, and there will be very interesting things to observe in this late stage. The problem is, as usual with these experiments, it takes you so long and it's so expensive to get there that you have kind of limited opportunities to do that. And a side note, as a consequence of what we have said, is that if you do these experiments twice, the outcome is, of course, slightly different. I mean, the general pattern is the same, but the fine structure is not the same. That makes a lot of sense because it depends on the non-shared environment part. And that means that if you would do such a study that starts later in life and looks at these effects only later in life, and you really want to pinpoint certain correlations, that you would have to do the study a couple of times. So N would be becoming the study rather than the individual mouse. And that's, of course, extremely challenging. And the question is whether that's really worth it and whether there are not lower hanging fruits that tell us a bit more. But you're absolutely right. This is it's a very good question.
All right, thank you for your answer. The next question is, do you start the travels of the mice so that they receive different stimulations at different times in their journey? And can you influence their behavior, their social behavior, their leadership, and their so-called followers by this stimulation path?
So, I mean, the questions, and that's really interesting. They go a lot in this direction of the social structure. It makes a lot of sense also to us. But again,
we're going to have you back because everybody's asking questions about the social.
To showing you this stuff. It's really exciting, but it's not finished. So I don't really tell too much yet. But you're pointing exactly at the right questions. And the issue of how I can manipulate these trajectories is really interesting. And we are doing a study on learning in this context, so where we expose the mice to learning. And then you have a hand on what happens in the environment, because you know if you give them a learning stimulus rather than just putting them in a cage, then it's better defined on what the mice respond to. Of course, this is not everything, but at least you know if they respond in the way that you expect them, you know that they must have seen the stimulus and respond to it. And then you have a grip on what you're describing, that you can play with these trajectories. So this is a very interesting line of research. But you can imagine it's also quite challenging in terms of the setup.
Maybe one more burning question.
Okay, so Professor Gerd, have you looked at the effect of population size? Does overcrowding, the stress caused by overcrowding, tend to be counterproductive in neurogenesis?
Oh, we haven't done that. We haven't done it. We have done a small study many years ago on group size. So, and there is to a certain extent, there's a correlation between neurogenesis and group size. So if you have more, it's a little bit the thought of the enriched environment. And larger groups, mice are social animals, so they love to be in larger crowds. But of course, there is a limit to that. But we didn't push the experiment to that limit, far away from that. So I would predict that, yeah, at some point this tips, and then the mice don't like it anymore. Then you get a negative effect. Yeah.
So Gerd, thanks for coming on the show. Thanks, Kalisha. We've given you about 20 more years of experiments to do tonight, so hope you're taking notes on that.
I certainly will write them all down, and then if we meet in 20 years again, then
And we'll definitely have you back to hear about the social effects. I want to remind everyone to use the chat function and panelists and all attendees option to leave comments and feedbacks on the show. I also want to point out a new study contributed by my co-host, Andrea Meyer, which aimed to identify specific white matter hyperintensities and the location associated with vascular risk and cerebral amyloid A-beta 42 positive status in patients attending memory clinics. Data from 11 memory clinic cohorts were harmonized with 3,100 patients. And white matter hyperintensity volumes in 28 regions were related to vascular risk, compound score, and A-beta 42 status. There's a link to that in the chat box if you'd like to read about it. I want to also leave you with our last video, which is about a woman in her 90s that's proving age is not just a number, or age is just a number, sorry.
音が良くなった音が良くなった音
Like sunshine, like springtime, like something's in the water and I'm taking a deep dive. I'm feeling so weightless, like I'm gonna make it, and nothing in the universe can take this. I can see it clearly now, nothing gonna bring me down.